don't think I do. All right, so this is a feedback form that you'll be using, right? So you don't need to put your name on it. In fact, I'd probably rather you didn't, to be honest, because it's good that these things are anonymous. That way you can write whatever you wish without any worries about how it might be interpreted or not. But you do need to put the candidate's name on. That's super, super important because obviously we need to know who you're talking about. Let's bring up the chat. Someone just said something. Yep, just give me a second, both of you, and I will hopefully answer all your questions. I'll keep them in mind, make sure that I do, okay? Pardon me. So get out of it. The actual forms for you to use are here. So this is in the exams and quizzes in the course content or in the Blackboard. Same form for both of them, so you only need to download it once. I just stuck it in both of them for easiness sake. Essentially, these are assignments where, if I've done everything right, hopefully, fingers crossed, you will simply click on this, get the form. The invited lecture is at the same time as normal class, so it will replace the class, or rather displace the class, because we still need to cover that stuff. We're just going to be shuffling things around a little bit so we make space for them. Right, so they will be in the normal class period, so this 9 till 9:50, but you'll not be using the same Zoom link. The Zoom link that you will use, let me see, it's in here somewhere. Here we go. So in the announcements on your Blackboard, you'll see the new Zoom links, right? So these are specific to those invited lectures. There's a different one for each of them, right? Because that's what's been done by the person that's organizing all of the Zooms for those candidates on those particular days, right? So rather than clicking on this one, I'm not going to be controlling it, right? It's going to be controlled by the administrative assistant to the dean. She's the one that's kind of organizing all this stuff, which is great because it means that we don't have to. But she's the one that's going to be getting it up and running. It'll be running and ready by 9 o'clock. Obviously, I think it starts. Yeah, don't worry about that. I'll just confuse you. But it'll be there. You just have to click on that and join, right? The deal is that I will be obviously in that Zoom as well because that's part of my job to assess the candidate. But I will be taking a note of attendance. So literally just as a checklist, like you're there, at least signed into the Zoom. And then ideally at the end of that Zoom, or, soon after, you need to fill out that form and upload it to that assignment. Where are we at? This one here, right? If you do both of those things, right, you attend the Zoom, right, and you fill out the form, you get 10 points in addition to your extra grade. So it's true extra credit, right? So that's for one of them. Repeat the same thing on Wednesday, different Zoom link, same form, same process. You get another 10 points extra credit. Those forms have to be uploaded by midnight the following day. Right, I'll just type that out. It should have been in the announcement. No, generally they're not covering material that we need to study, thankfully, because I'm not going to understand a bunch of what they're talking about as well. Right, bioinformatics isn't my specialty by any stretch. So no, it's just in the truest sense, it's a benefit to you to attend because you'll get to see some cutting edge research that are done by, our 3 top candidates. So it'll be cool science to actually hear about and see how people apply what we're learning to, real life questions. And obviously the benefit for us, for you attending, is to have your opinion and feedback on what you think about that candidate, because we have our own opinions, but there's only 5 of us on the committee, and we're professors, right? So we have a different perspective. And so getting your perspective as students, who will actually be the people that will be interacting with these new professors, most of all, really, in their professional capacity, is really, really important. Obviously, the benefit to you, in addition, is potentially 20 points towards your total for the course. And so 20 points works out to, it's a little bit less than what a homework's worth. Similar kind of amount of time, give or take. Or it's essentially equal to 1 grade division in an exam. And so even though it's not going to be applied to your exam grade, pardon me, directly, it will count as, you know, if you got, say, 70% in exam 1, effectively that boosts it up to 80%, which is pretty neat, I think. Does anybody have any questions? I think I covered the questions that I got in the chat. Yeah. Let me just type up the date. They're due midnight the day after. So, e.g., first Feb feedback is due 11:59, I think it is, PM on the 2nd. Right, so please don't forget to do that, because it shouldn't take much effort to do, and you need to do that to get the credit. Right, so you need both of those to get your 10 points, and I wouldn't want people to miss out. Okay. Actually, let me see. You know, I always tell people to use their calendar, right? So this is, I'm going to make myself a reminder to send to lecture, because I've got a terrible memory. Hopefully I'd remember, right? Because, you know, I'm getting better at stuff like that. But day reminder, awesome. Okay, cool beans. Cool. Anybody got any questions? This is how I remember everything that I'm supposed to. My wife's going away for a couple of weeks, so I'm going to be putting all of the kids' stuff in there as well. It's going to be pretty crazy. All right, so let's crack on, because I want to get through Mendelian genetics today at the very least, so that we have next Friday and after that to do the probability and statistics stuff. Right, and it's actually going to be a little bit tricky next week for homework 1, because you're going to need to know how to do a chi-squared, and normally we'll be covering that in the second lecture on probability and statistics. So remind me next Friday, because again, I've got bad memory, to cover chi-squared first. That way you won't be lost at sea on the homework, which is due, I think, the Saturday after. I'll try and remember, but if I don't, remind me, you know, because I might forget. Okay, so let's just ever so quickly go back to this. So we finished up the monohybrid cross on Wednesday with our Pomeranians and Schnauzers, and where we are. All right, and so what we need to do now is finish up 2 things. 1, what is a test cross, right? Which is, you know, kind of laid out here in front of you. 2, we need to go through the dihybrid cross. Make sure that that all makes sense to you, okay? And so the dihybrid cross really isn't too tricky. It's essentially an extension of a monohybrid cross, right? The test cross is quite different, right? And so these all have very particular names because when we use that name, that tells you we're actually talking about something very specific, right? And so let me get my whiteboard because we'll be needing this. Make sure I don't snag my laptop cable. So here's a question to class. Which phenotype for a monohybrid cross, let's say, that applies to any dihybrid cross too, but which phenotype can you tell definitively with 100% guarantee what the genotype is?
Recessive.
Yay, well that was quick. Why?
Because if it doesn't appear, then you would assume that it's a recessive genotype.
Okay, but what do you need for the recessive phenotype to be seen? Just trying to get you to think through this in different ways. One, you need an absence of a dominant allele, right? It's one way of thinking about it. Because the dominant allele will mask another recessive allele. Or another way of thinking about it, you need to have only recessive alleles to see the recessive phenotype, right? So that's the only phenotype that you can tell the genotype for. What if you need to figure out what the genotype of a dominant phenotype individual is? How would you do that? Right. The best way of doing that would be to cross that dominant individual to a tester of a known genotype. Right, so let's turn this around. Oh, got to close the curtains. Be back in a second. It was a little bit darker than normal today. Anyway, all right, so you can see this around, so parallax isn't quite so bad. All right, so we have a unknown, right? So we have, let's try and write this up. Essentially, there we go. Our dominant individual could be either homozygous dominant or heterozygous, right? We don't know which because we only need one dominant allele. Oops, that's not very good. One dominant allele to be able to see that dominant phenotype, right? So what do we have to cross that individual to be able to see a recessive phenotype, to be able to figure out whether or not we have a recessive allele?
You would cross it with a recessive.
Oh, I'm so sorry. I forgot to put my headphones back on. That's stupid, wasn't it? Sorry, say that again.
You would have to cross it with a recessive to see if a offspring is born with the recessive trait.
That's right. Thank you for that. Sorry, I heard this like tinny little tiny voice speaking. I was like, oh, someone's speaking to me from my couch. And then I realized I hadn't put my headphones on. All right. So you need your tester, correct? And that has to be a homozygous recessive, right? And so what would you get if you crossed a homozygous dominant individual, or let's say your dominant individual was homozygous dominant, what offspring would you get? So let's say our mystery was homozygous dominant. What's that going to give you?
You would get heterozygous dominant, wouldn't you?
That's correct. You don't need to say dominant with heterozygous, just heterozygous is fine. But yes, that's exactly right.
Okay.
All you would get, because this one is only going to produce alleles with the dominant allele, not alleles, gametes, sorry. And this one's only going to produce gametes with the recessive one. And so you'll get 100% heterozygous, right? So let's kind of cross that there. What about if your mystery individual is actually heterozygous? What are you going to get in the offspring now? Oh, actually, let me back this up a little bit. What gametes are you going to get from that heterozygous individual?
Both dominant and recessive?
That's exactly right. So you're going to get both, or you're going to have gametes with either a dominant allele or a recessive allele. What's going to be the ratio if the law of segregation is followed of one to the other?
Is it 1 out of 3 is going to have the dominant heterozygous?
That's a good guess, but no. So what does the law of segregation tell us? That alleles segregate into gametes randomly, right? So there's nothing that changes which allele goes into which gamete, right? So if we have two different alleles and their segregation is random, what ratio would you expect? Think of it like a coin toss in a sense.
50-50.
Yeah, exactly. It'll be 50-50. So it'll be 1-to-1 or 50-50 ratio of those two, right? So if we have 50% of the gametes from this individual having a dominant allele, 50% having a recessive allele, what are we going to get in our offspring?
You'll get hetero and homozygous?
That's right. So we're going to get that and that. And what's going to be the ratio?
1-to-1.
Right, kind of already written up. It's exactly the same as the ratio of the gametes, right? So we're going to have 50-50 or 1-to-1 or 50% or 50%, whichever way you want to spin it is totally fine. So really, actually, the numbers, they're important to understand because they're kind of central to how this thing works. But really, all you need to look for are homozygous recessive individuals. Because the only way that you'll ever get even one of those, right? Even if something funky is going on, even if you only get one of those, that tells you this individual, this unknown, has a dominant recessive allele, right? So it has to be heterozygous. So that, in a nutshell, is a test cross. It's really the only way, short of sequencing the genome of that organism, that you can actually determine whether or not a dominant individual is dominant, homozygous dominant, or heterozygous, is by doing this. Does anybody have questions? Yeah, my power cord doesn't stretch quite far enough. No? Okay, so almost certainly I will give you at least one question based on this in the exam. I might be giving it to you in terms of the output, right? So we have an individual with this particular phenotype combination, and when it crosses with one with this, for example, we get these particular offspring out. What is the genotype of the parent, for example? Or, you know, variations on that kind of stuff. So you need to be able to understand kind of what that means. Where's my camera gone? So that you can answer those questions, right? Those are going to be good examples of those kind of higher level, more difficult questions that you'll see on the exam. There we go, not too blurry. It's a downside of doing this stuff in the dark. Okay, let's get this off so we have it free for anything else. Cool. Okay, so water. Okay, let's move on to dihybrid crosses, right? Which are essentially, sorry, I've got to take my jumper off. Or sweater, as they call it in the US. Here you go. Here's a funny joke. Well, I think it's funny. You have to be kind of British to appreciate it. If this is a jumper, what happens if you cross a sheep and a kangaroo? Yeah, woolly jumper. Get it? Anyway, that's a British dad joke. I'll try and get some American dad jokes. Oh, here's another one. What does the buffalo say to his son when he goes out to work in the morning? Bye, son. That's one of my favorites. Anyway, let's get back to business. I've got loads of those. My dad sends them to me all the time as well. So, dihybrid cross, pretty straightforward in many ways. There's a little twist which will potentially get you caught out, but let's get this back down here. Similarities and differences, right? So in terms of similarities to monohybrid cross, pardon me, we have exactly the same structure and exactly the same assumptions. So we have our homozygous parental generation, right, where we're crossing two true breeding parents together, one homozygous dominant, one homozygous recessive. We have our F1 generation, which will always be heterozygous. And as such, oh, here, who remembers this? What phenotype will they show? Which of the parents will the F1s look like?
Dominant.
Correct. Awesome. Got to be just like that, right? So exactly the same as with the monohybrid, right? And those will be crossed together, those heterozygotes, to produce our F2 generation, right? So this is all the same, right? The assumptions are the same, the structure is the same, the at the very least, the P and the F1 generation are the same, kind of, right? The differences are we have two genes, right? So we have two genes, each one has two alleles and two phenotypes, right? And so as a result, the F2 generation isn't going to be a simple 3-to-1 ratio. Now we have 4 potential phenotype combinations, right? Or we will see 4 phenotype combinations. The offspring can be one of those 4, right? We're going to see 2 phenotype combinations that are the same as the parents, right? So the dominant, recessive. And we're going to see another 2, which are a mix of those parents, right? So we're going to have parental phenotype combinations, and we're going to have non-parental or recombinant phenotype combinations. Okay, does that make sense? It seems a little rude to dive straight from dad jokes into dihybrid crosses, but sometimes that's just the way it rolls, right? Okay, so that's essentially one of the big differences. And the second one is that as a result, the ratio here in the F2 is going to be a 9:3 to 3:1 ratio, right? So that's really important. Oops. Right, as a result. And the 3:3, the two in the middle, are those non-parental combinations of phenotypes. Okay.
What's the ratio for the F1 generation again?
All heterozygous. 100%. So if you think about, actually it's very much the same as the test cross that I just unfortunately erased. Anytime you cross together a homozygous dominant and a homozygous recessive, the only genotype you'll ever get is heterozygous. Essentially because each parent contributes only one possible gamete, only one possible allele to the F1 generation. And so it's always heterozygous. But again, that applies very specifically to our mono and dihybrid crosses. Doesn't necessarily apply to other types of crosses, like a back cross or a test cross, or whatever else we might be looking at. Okay, so let's do this first. We're doing okay for time. We should definitely finish this up. Maybe if we get time, we'll crack on into a little bit of the probability stuff. I'll try and keep a rein on the tangents so we, stay focused. It's Friday after all. It's always difficult.
Quick question: are you? I've looked through the PowerPoints. Are you just assuming that we know how to use Punnett squares and all that?
Yes.
Okay. I just wanted to make sure I was right in assuming that.
Yeah. I'll quickly in a second go through the outline of the Punnett square that we'd be doing for a dihybrid cross. I didn't do one for a monohybrid cross because it's relatively straightforward. Actually, quick joke. I think it was an XKCD comic, if you've ever seen those, written by like some science nerds. It was basically a picture of a drawing of what happens when two geneticists go on a date, and it was like they're using a napkin to do a Punnett square of what their children would look like. I always think it's funny. I need to print that out and put it on my office door. Anyway, so yes, that is kind of an assumption, but I'll actually show you kind of how we would do that for dihybrid cross in a little bit. So again, we have our stereotypical, I guess one could put it, P generation. We almost always give you these as dominant, dominant, and recessive, recessive, right? Because it just makes it a little bit easier to grasp, but it doesn't actually make any difference, right? The only thing that would be different is what would the parental phenotype combinations be, and which would the non-parental ones be, right? Because if you cross a dominant dominant with a recessive recessive, you're going to get heterozygous individuals. If you cross a dominant recessive recessive dominant individual together, what are you going to get? Heterozygous individuals, right? You're going to get exactly the same thing. It doesn't actually matter. But for clarity's sake, we almost always give it to you as dominant, dominant, recessive, recessive, right? So now we have our two phenotypes. Actually, we've got two traits because we've got two genes. We've got seed shape, a lot of S's there, and seed color, right? And so our individual phenotypes are round versus wrinkled, yellow versus green, right? Actually, hopefully, later on, I need to do some reading up on this. I'm going to use leopard geckos for some genetics because those are super cool. I learned about those this summer with my daughter. Anyway, so which I'm kind of excited about. Kind of want to get to that point because it's going to be cool. But we have peas for now. And we're going to be generating, here we go. What are the F1s going to look like? Are they going to look like these? They're going to look like these. What phenotypes will our F1s have? I've already told you they're going to be heterozygotes.
The yellow round.
Yeah, exactly. They're going to show the dominant traits of the dominant parent, right? So now we have a much more complex question, right? What are the F2s going to be? And so these are our F1s. I'm going to write this out. This is still light enough, right? You can see what I'm doing. I'm trying to keep glare as minimal as possible. Can you see? How many fingers am I holding up? Come on, quick. Anyway. Sorry, I'm getting Friday fever. Awesome. Yeah, I haven't lost any fingers. Even though I keep trying, I still have all of my digits attached. Okay, so we have our F1, right? Our F1 is going to be heterozygous. Let's see if we can get this as straight as I can. There we go. Kind of constrained by how long my laptop power cord is, which is kind of funny. Okay, most important question of the day, what gametes can we get from this individual? Remember what I did for the test cross? I showed, if you've got this particular genotype, these are the gametes that you can get, different alleles in those gametes. What do we get from our double heterozygote? Just shout them out. I'm not even going to tell you how many of them. Let's see if we can figure that out, because that's a really important point.
You get one of each of them, right?
Well, you have to have one allele of each gene, right? So you're going to get an allele from this gene, and you're going to get an allele from this gene, right? So for every gamete, you're going to have one allele of each gene. That's right. But what different combinations of alleles are you going to get? First off, how many different combinations can you get? Let's start with that question.
4.
Yeah, that is totally awesome. Sorry, I don't even know who's speaking. This is super weird. Who is that?
It's Amber.
Oh, hey, Amber, that's good. I'm sorry, this is kind of like the weird thing about teaching online is that you don't actually get to see who's talking at me. So my apologies if I don't recognize you straight up. And yes, is that Jim? JM? I'm going to call you Jim. Hope that's right. Oh, awesome. Good. So yeah, you get 4 because you got 2 by 2 combinations. So you have 2 by 2, 4 potential combinations, right? And this is where it gets a little funky. And this is kind of the bit that essentially this is the point that can trip you up in a cross with 2 genes, right? You have to think, what are all of the possible combinations, right? Because you could have big R, big Y, right? You could have little r, little y, right? So essentially you can have the same gametes that you would from the parents, right? We don't actually give these a name, but they're essentially the same gametes that you would get from the original P generation parents. But you can also get a new mix, right? You can get a large R and little y. This gets a little bit complicated, but hopefully you're following what I'm doing. And the opposite, the converse, right? You can get a little r and a big Y, right? So these are the 4 possible gametes that you can get. Oh, JM, I'm sorry. I didn't realize. I've got a real small laptop screen, so I wasn't sure if my eyesight isn't as good as it used to be. I didn't know if I was missing an I between the J and the M. Or, you know, I'll call you JM in future. So if you get this from one parent, 4 different gamete combinations, what are you going to get from the other parent?
You would also get 4.
That's right, because both the parents are exactly the same. Right? I'm just going to take a quick pic of this so we can write that out. I have to deal with the glare from my door. It's ridiculous. I hope my bosses appreciate the trials and tribulations of teaching from my couch. Okay, so essentially this is where Amber, that's right, Rogelia, the same number. So this is where the Punnett square comes in, right? And I'm not going to go through all of this because I'd actually like you to do that yourself. But I'll show you how we lay it out, right? So we have one parent across the top, one parent going down the side, and we have the same possible gametes. Now, again, going back to what we talked about, the test cross, what proportion or ratio would you expect to find these gametes in?
One to one.
Keep going. That's right, but you left out 2 extra numbers. 1 to 1 to 1 to 1, right? Because there's 4 different combinations. But yeah, they are equal. They're equal because of 2 different laws, right? One, the law of segregation, right? So the R gene, right, will segregate its 2 alleles, the big R, little r, equally, right? So we have right, 50-50 chance of getting a big R or a little r. Same applies to the Y gene. 50-50 chance of getting a big Y or a little y, right? And the second thing which results in us getting a 1 to 1 to 1 to 1, I think that's right, numbers of 1, ratio is the law of independent assortment, right? So whatever allele you get for the R gene, whether it's a big R or little r, has absolutely no effect on which allele you get for the Y gene. That essentially is the law of independent assortment. And so those are really, really, really critical assumptions. And we'll see later on how those assumptions can get broken, particularly the law of independent assortment, right? And I'll touch on that a little bit when we finish this, just to kind of put a worm in your ear, so to speak, right? And because we have the same parents, we have the same combinations. Sorry, I kind of lost the double crap sticks. I need to get an extension for my laptop. I can't move it far enough for you to see the whole screen. All right, but if you're missing something I'm saying or writing, just give me a shout and I'll repeat it or move it. And so essentially here's our Punnett square for our dihybrid cross, right? So if you got 4x4 different gamete combinations, how many different outcomes can you have in terms of genotype? Right, times 4x4, you're going to get 16, right? Yeah, totally. So, but we don't have 16 different phenotypes. We have 4 different phenotype combinations, but that 16 is going to be important because that's essentially going to inform our ratio, right? And so all you do is you just add these alleles together, right? So that gets you a HOMDOM for both of them, right? Which is going to be, what's that, round and yellow? And you repeat that for all of them. So all of this line are going to be the parental dominant phenotype. All of this line will be the same, right? Because we have at least one dominant allele for each. Genotypes will be different. I've never actually worked out a genotype ratio for a dihybrid cross because it's just a little bit too crazy. But the phenotypes will be the same, right? Now over here, right, we have recessive, 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 recessive. This one here, all on its lonesome, that is our sole only parental homozygous recessive individual, right? That's our 1 in 16, right? Because we've got 16 squares, we're only going to get one of those. That's our 1 at the end of our 9:3:1 ratio. We're going to have 9 of these. Where do we have to go to get? Here we go. For example, this would be because we have recessive R up here, doesn't matter what the Y is, recessive R up here and a dominant Y. And so we get a homozygous recessive and a dominant allele for Y. Doesn't actually matter whether this is 2 dominant Ys or not. As long as there's one there, we'll see the dominant phenotype. Right, so this one will be wrinkled and green, wrinkled and yellow, sorry. So this one is going to be one of the 3, well, one of the 6 essentially, non-parental combinations out of the total. We're going to have 3 of each, right? So try and do this for yourself. It's good practice. And when you do, just go through them all and check each individual square and go, okay, do I have, there's my hand, dominant for R? If I do, it's yellow. Do I have dominant for Y? At least one. If I do, it's round. Hang on, have I got that right? Yeah. Got them actually crossed up. But you know what I mean. As long as you have a dominant for each, you'll have the 2 dominant phenotypes, right? You should get 9 of those, right? And just go through those and you should get our 9:3:3:1 ratio. We're going to get 9. So this is going to be the dominant parent, right? This one is going to be the recessive parent. And these 2 are going to be non-parental, right? Because those will have a phenotype. Yeah, totally, Rogelio, you got it dang straight. That's exactly where we get the 9:3:1 ratio from, right? The 16 total, right? So essentially 3/16 of the offspring from this cross will be, let's say, wrinkled and yellow, right? Only 1/16 will be wrinkled and green, right? That's where these numbers come from. Does anybody have questions? Questions, questions, questions. Okay, well, go through this yourself. It's good practice. It will kind of give you some understanding of, it's just like muscle memory, essentially, right? Doing these things. Now, one thing to put a worm in your ear, right? If this ratio changes from a 9:3 to 3 to 1, and when we look at doing a chi-square, we'll see, you know, how much that needs to change for it to be interesting. But if it does change by an interesting amount, a significant amount, that's going to tell us something about what's going wrong with our assumptions, right? So one of the classic things is this ratio might be like a 12 to 3 to 1 ratio, or it might be, what's the other one? I think it's 9 to 4 to 3. But if you get a different ratio here, that could tell you either these phenotypes are interfering with each other, right? Which is a phenomenon called epistasis, right? Or, so that's one potential hiccup that can occur. Or it could tell us, particularly if we see fewer of these than we should do, right? So, or another way around, if we see an excess of these parents, what that's telling us is that our alleles, our genes, are not assorting independently. So that tells us that our law of independent assortment is not being followed in this particular cross. And that's called linkage, right? Where two genes are close enough together that they affect the inheritance of each other's alleles, right? We'll get to that. Actually, we've got, I think like 3 lectures penciled in for that, because that's a bit of a doozy, right? So it's fun if you like kind of math problems. If you don't, it can be challenging, but I'll be spending a bunch of time with you going through that, right? So if this ratio differs, like what you get from what you expect, you would expect a 9 to 3 to 1 ratio. If you get a different ratio, then that's telling you that something's going on. One of your assumptions is not correct, right? And so I want you to just, come on, keep that in mind. It's got, bloody hell, I'm getting glare from my laptop now. That's ridiculous. Okay, so we've just got a minute left. And so all I want to do now is I want you to think about this as what about if we do this as a test cross, right? So kind of essentially getting you to think a little bit kind of outside of the box. What if we were to take a unknown individual, right, expressing the dominant traits, dominant phenotypes, 2 of them, because we're going to be dealing with 2 genes. What ratio would you expect? And we're going to use our homozygous recessive tester. What ratio of offspring would you expect if your unknown was a double heterozygote? So remember the test cross, we're going to be crossing a unknown with a homozygous recessive. What ratio of phenotype combinations would you expect in the outcome of that cross if the unknown is heterozygous for both of those? I think it was Amber that gave me the ratio of the gametes. that you get from a heterozygote? Almost, not quite. Think through what if we have allele segregated normally, if we have genes are sorting independently, what ratio of these gametes do you get from a heterozygote? We get 4 different gametes. Are these going to be present equally or not? No, almost, but not quite. We're going to have a 1 to 1 to 1 to 1 ratio. That's all right. So the reason is this individual doesn't really matter. All it's doing is making sure that we see the recessive allele in our unknown, right? And so we will see a recessive allele here, right? So this individual will have double dominant. This will be, what's that going to be? Round and green. This will be wrinkled and yellow. This will be wrinkled and green. And because these alleles are segregating independently and randomly, we'll get a 1 to 1 to 1 to 1 ratio. All right, I think that's enough for today. Just to put that in your head, right? And those are some of the kind of more advanced questions that you'll be getting in the exam, right? To really kind of make you think about stuff, to really test whether you understand the principles that we've been learning. Cool, I think that's more than enough for a Friday. So I'm going to sign out. Have a fabulous weekend, everybody. And see you on Monday for the invited lectures. Again, I'll be sending those links out again on the night before, right? Just as a reminder, so you have them to hand. Cool. See you later, everybody.